Hello everyone, welcome to Artistic Atom channel. I am Fatima and today we will be discussing about the scientific research method. This is the part 1 of this session. In part 2, we will be solving a task sheet based on the same concept. Now, at the end of this session, you will be able to explain the scientific method in your own words. Nextly, you will be able to describe all the steps involved in this method. And lastly, you will be able to apply this method to different problems. Now, when we talk about a scientific method, it always starts with a question or a problem. Now, you all might have this question in your mind that how come we get a question or a problem to start with a scientific method? Well, we always get a question or a problem from our daily life experiences. Now, when you are observing something, you might get a question like why this happens. If suppose you're confused with something, then you will start thinking that how I'll be able to solve this and what could be the answer for this. And suppose if you're interested in something and it brings up you with a question like it sparks a curiosity in your mind, then also you will end up with a question. So when you have these observations, you will end up with this thought that you start to find out what are the different ways or how these things happens or why they are happening. So with this you end up with a question or a problem. Like for example, if in your garden you're walking and then you have observed your plants, you see that these plants, they're growing in different directions. So that sparks a curiosity in your mind that why these plants, they're growing in different direction, what could be the reason. So here's how you end up with a question. Okay, now when you have got your question, the next step starts. It is the process of investigation, where you're trying to either find out the answer or you're trying to produce some or the other product so that you can solve the problem that you had at the beginning. And lastly, once you have got your specific data to find the answer, or you have produced something to resolve the problem, you will look for the measurable reliable results. Why? Because these reliable results will help you in answering the question that you had at the beginning. Okay, so in conclusion or in a nutshell, what we can conclude is scientific method starts with a question, then you starts to investigate and try to find out the answer for that question. Now once you have got that particular answer, you're not even sure of it, so you starts to check the reliability of that result. And once you are satisfied with the results that you have got with a concrete justifications or the proof, you will get a proper answer to the question that you had at the beginning. So this is how actually we carry out scientific method. Let us understand this in detail. So, the next question that you might have in your mind is who actually uses this scientific method? So, well, everyone uses it in their day-to-day life. They, in order to solve these problems. But the only thing is they are not even aware that they are using scientific method. But yes, we do use it in everyday life. Now, what is actually this scientific method? So it is nothing but a systematic way with which we solve a problem. So let us see what are the different steps involved in a scientific method and then see a question or a problem where we apply the same thing and see whether we are able to solve it or not. Now, the first step in scientific method is observation. As we have discussed earlier, that when you're observing something or when you're curious about something, it will lead you to a question or a curiosity where you will try to find out the answer to the question that you have in your mind. So, like for example, we have taken this example at the beginning that when you're observing your plants, you see that they grow in different directions. This will spark a curiosity in your mind and then you will start to think that why this is happening. Okay, so the first step is observation, from where you will get a problem or you will end up with a question. So once you have got your question, then you start to frame it, that what makes these plants grow in certain direction. So firstly you have observed, secondly you have framed your questions. Now nextly you will start doing research. So in research, what do? In research, we starts to gather the information. So From where we get this information? Either you can get it from books, magazines, reports, or you can ask experts. Even your past experiences also provides you with different information. And also if you have some prior knowledge, that will also give you a lot more information about the question that you had at the beginning. So this is how actually we carry out research. That is, we starts to gather the information. So if we apply this to the question that we had at beginning, so this could be the research conclusion, that when we have learned about different plants and also different processes that they follow in their life, we end up with this concept of phototropism. Then when we study about this more in details in our research, so we end up with this particular thought, that is, the plants, they respond to light sources. So we end up with this conclusion, or we have gathered this information about the question through the research, that is nothing but plants, they are directed towards the light because of phototropism. Hence they grow in different direction. This is what our beginning thought is. Now once we have got our beginning thought, we starts to a tentative we starts to frame a tentative answer for the question that we had. So always this hypothesis starts with the word I think. Okay, so when we use this word I think, we frame our hypothesis like this, that I think the direction of the plant growth depends on the location of the light source. So this is the temporary answer. This means you're not even sure of it, but through your research, this is what you have understood till now. Now, in next step, what you will do? In next step, you will conduct an experiment where you are actually trying to test your hypothesis, whether whatever you're thinking or concluding after research is actually true or not. So this in your experimentation, it is nothing but a series of tests that you carry out in a controlled environment. Now why we are carrying out this experimentation? Obviously to validate our observations, that whatever we have observed is actually true or not. Okay, and also to get an answer to the question that we were having at the beginning. Now, here I have used a term called as controlled environment. So, what do you mean by this term? Controlled environment is nothing but the surrounding conditions. So, when you're carrying out those experimentations to prove your hypothesis, always make sure to keep the surrounding conditions same. Like, for example, amount of light that is entering, heat, or we can say temperature of that particular room, or the humidity, etc. So all the surrounding or the conditions in that particular room where you're carrying out the experimentations are called as controlled environment. So always you're going to carry out your series of steps or those tests that you have planned in experimentation in a controlled environment, so that they are not affecting the results and the results are conducted fair. Now the next is, when we talk about experiments, always there are variables. So what do you understand by the term variables? So variables are nothing but that can change by themselves, or we can change them in an experiment. We term those factors as what? Variables. Now there are three types of factors. First one is independent variable. So what do you understand by independent variable? Independent variables are nothing but the factors that you can change on purpose. Like for example, when I'm conducting the plant experiment to find out why these plants, they are growing in different direction, at that point of time I am changing the direction of light to see whether they will bend towards the light source or not. So this direction of light is nothing but my independent variable, because I am changing it on purpose. Now the next one is the dependent variable. Dependent variable is nothing but the response to the independent variable. Like for example, when I'm changing the direction of light, so what is happening? The plant is growing in the direction where there is light source. So the direction of growth of that particular plant is nothing but a response to the independent variable. So I will call that particular growth factor as what? Dependent variable. Hope you have understood it. Now, the last factor is the controlled variable. Controlled variable is the factor that you are keeping same in the experiment. So, whatever are the conditions or the factors in the experiment, you're keeping same. They are called as what? Controlled variable. Now, here I have used this term control variable. You might have this question in your mind that if the control variable doesn't change, then why I'm calling it as what variable? Because variables are what? They change by themselves or we change it. So if we are keeping control variable same, then why we are calling it as variable? So even though the controlled variable is called as variable and it doesn't change throughout the experiment, it is not changing. Why? Because we are keeping it same. Okay, and if we do not keep it same, it will change by itself and will affect the results. Hence they are called as what? Variables. Okay, so why control? Because we are keeping it same. So suppose if we do not keep it same, they will change by themselves and affect the results. So I told you at the beginning, only variables are the factors that can change by themselves or we can change it. So it is changing on its own, hence we call it as variable. And since we are keeping it same throughout the experimentation, we call it as what? Controlled. Okay, so hence the in conclusion, the name is controlled variable. Hope you have understood this. Now, the next question that you might have in your mind is, what is the difference between controlled environment and controlled variable? Well, it is very simple. When we talk about controlled environment, it means the surrounding conditions in the room or the place where we are carrying out the experimentation. Like, for example, temperature of the room, the light amount that is entering inside the room, humidity of that particular room. These all are the surrounding conditions in that particular room. So we call them as what? Controlled environment, because we are keeping them same throughout the experimentation. And what is control variable? Control variable are nothing but the factors in the experiment that we are keeping same. Like for example, when we are talking about plant growth in different directions, so there we are keeping the type of soil as same. Okay, and the pot in which the plant is kept, that is also same. Amount of water that we are adding, so whatever are the factors that we are keeping same in the experimentation is called as control variable. Hope this clarifies everything. Now, let us understand all these terms through a proper experimental process so that we are clear about everything. So even if you have a question in exam, you will be able to make out all the factors easily. So we will take the same example that we took at the beginning. So once I was walking in my garden and I have observed different plants. What I have seen that these plants, they are growing in different directions. So this is my observation. Now, based on this observation, I was quite curious that why this is happening. So it led me to ask a question that what makes these plants grow in certain directions. So with this, I have end up with a question. Okay. So first I have observed the situation and then I am ending up with a question. Now the next step is conducting a research so that I can find out what is the exact reason behind. So when I have conducted research, I was going through different books, different magazines, papers, research papers, review papers. So I have got the conclusion that there is a phenomenon called as phototropism, which actually makes these plants grow in different direction. So after this, I have framed a statement like I think that the phenomenon that I am observing in front of me is because of the same, but I'm not sure of it. So this statement that I have framed is called as a hypothesis. Okay, so it is the statement which uh, you know uh, pens down my thoughts about the research that I have done over the question that I was having. So after this hypothesis, I'm not sure, but I know that this might be the answer. So I have to check my answer and find out the reliability. So for that, I'm conducting an experiment. So when I'm conducting an experiment to test my hypothesis, I start to set up an experiment. So here what I'm doing, I'm using the identical plant placed inside a pot in a controlled environment and also in a laboratory room where I'm keeping all the temperature, everything same. Even the amount of water that I'm going to add is same. Soil type is also same. Humidity is also same. Okay, Now what I'm changing in the experiment is the direction of the light source. And after all these setup, I'm going to record all the observations. Okay. Now from this whole experimental setup, let us determine the following things. Firstly is the controlled environment. So whenever you're trying to make out what is the controlled environment in your experimentation, always ask yourself this question, that where is the experiment happening and what conditions of the surrounding are being kept or the same throughout the experiment. Those conditions are nothing but your controlled environment. Like for example, temperature of the room, amount of the light that is entering the room, humidity, airflow in the room, and also there should not be any disturbance. And we are using the same lab for throughout the experiment, we are not changing the lab room. So these all are the surrounding conditions, so they are called as the controlled environment because we are keeping them same throughout the experimentation. Now, the next one is controlled variable. So, whenever you are trying to determine the control variable, ask yourself a question. What specific thing or a factor inside the experiment you must stay same so that it is not affecting the independent variable? Okay. So, these are the factors that you're going to keep same in the experiment. Like, for example, same type of plant we are taking, the amount of water that we are adding is same size of pot we are taking, soil type is also same. Now the time for which we are keeping these plants under sunlight is also same, that is duration of exposure, and the instruments that we are using for all the plants we are keeping it same. So these are the things that I'm keeping same in the experimentation, so I will call it as what, controlled variable. So, in conclusion, easiest way to remember the difference between controlled environment and controlled variable is as follows. So, controlled environment is the place where you're carrying out the experimentation plus the overall surrounding conditions of that particular room. And in case of controlled variable, it is nothing but the specific factors that you're keeping same in the experimentation. Okay, so remember these two key points. Now the next is independent variable. So remember, whatever you are changing or you are manipulating, remember this word manipulate, because I'm going to give you a trick to remember all the variables using this term. So whatever you're manipulating or changing by yourself is called as what, independent variable. So in this experimentation, what we are changing? We are changing the direction of light. So this factor is nothing but my independent variable. And whatever is the response because of this independent variable is called as the dependent variable. Like for example, here I am changing the direction of light. Once it was in left hand side and other time it was on the right hand side. So I can see that when I have changed the direction of the light, the growth of the plant direction has also changed. Earlier it was in the left hand side direction and later it was on the right hand side direction. So here the direction of light source is the independent variable, and because of this the direction of plant growth has changed. So that is the response because of the independent variable. So this response is called as a dependent variable. Hope this clarifies about all the variables in an experiment. Now here is the ultimate trick to remember all the variables, and also this trick will help you when you are plotting a graph to decide which factor will go on which axis. So it starts like dry mix CC, where D stands for dependent variable, R stands for response, and always this factor you will plot it on y-axis. M stands for manipulation, whatever you are changing. I is then this particular factor is called as independent variable, and always you're going to plot it on x-axis. Now CC is nothing but constant control. So whatever you are keeping constant, or we can say same in an experiment, you will call it as what, controlled variable. So let us see how to apply this to the question that we have seen earlier in previous slides. So in previous experiments, what we were doing, we were changing the direction of the light. So when we were changing the direction of the light, that was our manipulation. So direction of light was our manipulation, hence we will call it as what? Independent variable. And if I'm plotting a graph based on that data, I'm going to plot this factor on x-axis. Now because of this manipulation, what was the response? The response was plants were growing in different directions. So the direction of plant growth is nothing but my response, and I will call it as what? Dependent variable in that experiment. And if I'm going to plot a graph based on that data, I'm going to plot it on y-axis. And whatever are the conditions in the experiments, I have kept same, like the amount of water, the size of pot, type of soil, everything was constant. And these variables are called as what? Controlled variables, because they were kept same throughout the experiment. Hope this makes you understand everything clearly and all the factors are clear, and you will be able to apply this trick to the question. Now, once you have got the data, after experimenting, you start to analyze the data. So when you start to analyze the data, what exactly you do? You start to gather the information, you start to document the information, and also make it in a way, or document it in a way that if others are reading it, it makes sense to them and they will be able to understand what was the exact problem and what could be the solution to it. Okay. Now this data you can gather or record or document in the form of charts, graph, or written works. Okay, so this is how actually we carry out the analysis, where we start to document our data. Now, once we have documented our data, we draw a conclusion from an analysis that this could be the tentative answer for the hypothesis that we had at the beginning. Now, sometimes it might happen that whatever is the conclusion might not align with the hypothesis that you have, and it might not correct. Your hypothesis might not be correct. So it's okay. You don't have to worry. The reason behind is because sometimes it happens that your hypothesis is incorrect, but whatever is the conclusion that you're getting is teaching a lesson to you that this is the correct concept that you should follow. So you will going to learn every time whenever you're carrying out a research, because you will end up with the correct information. So sometimes it clarifies all your queries and gives you the correct answers. Now, once we are done with the conclusion, scientists starts to perform, repeat their experiments, and that process is called as what? Verification or checking. What do they do exactly? Here they repeat their experiments again and again with the same material and same procedures, same conditions. Why they are doing this? To check the validity, so that if some other people or person or some other scientist is conducting the same experiment, they will also get the same results, so that their data and their research is valid and others can use it for their benefits. Now, so in conclusion, this is how actually a scientific method looks like. It consists of these steps. First, you're going to observe, then you will get a question or the problem, then you will go for the research and you will try to gather the information or the data. With that information and data, you hypothesize statements like, I think this could be the answer. But since you're not sure at this point of time, you carry out an experimentation to prove your hypothesis. Once you're done with your experimentation, you start to gather the data and start to document it and analyze everything. And after analysis, you finally conclude everything. This conclusion will give you the answer for the question that you had at the beginning. Now, once you're done with the conclusion, you start to verify things, whether these procedures are reliable or not. So suppose if others are also conducting the same experiment, they should also get the same answer. So it is necessary to check the validity of the results. Now always remember, scientific method is a complex and flexible process. It doesn't mean that always you have to follow these steps step by step, because sometimes what may happen in an experimentation, scientists feel like that this step is not necessary. So they can skip or they can manipulate or change the step in order to get the best results. So it is not necessary to strictly follow these results. It is just structure or the scheme of work that you should follow when you're carrying out a research. But suppose if sometimes some skipping some or the other steps or switching them gives you better result, it is allowed. You can do so. Now, once scientists are done with their whole research, they share with the world. So, when they are sharing it with the world, what happens? Others also learn from their mistakes that they have done, and this research also helps humanity to make it better, and also provides them with a different scope for the future studies in that particular field. So it provides knowledge to the others also. So once they are done, they share their information and research whatever they have got. So with this, we have completed our session and we have learned everything about the scientific research method. And I hope you have understood everything. In the next session, what we will be doing, we will be solving a worksheet based on the same, so that you're clear with the concepts, and also you're able to apply the concepts to the questions. Thank you so much for watching. See you in the next session. Like, share, and subscribe to Artistic Atom channel. If you have any queries, you can also comment it in the comment section. Thank you.